in neotropical Pacific birds.
All right, great. Thank you everyone for coming. Good morning. Again, my name is Nick Mason. I'm a PhD student at Cornell. And today we'll be talking about some macroevolutionary patterns and avian vocalizations across two groups of neotropical passerines. I'd like to start by thanking my collaborators, including Kevin Burns, Joe Tobias, Nat Seddon, Liz Derryberry, and Sam McClarmont. Kevin and Joe are both here, and I encourage you to check out their talks on Monday and Sunday, respectively. And so one of our overarching goals in evolutionary biology is to understand patterns of biodiversity, both with respect to species richness as well as phenotypic diversity. And one kind of anecdotal observation about biodiversity on the tree of life is that it is heterogeneous with respect to space and the lineages on the different trees of life. There are some lineages that have lots of species and lots of phenotypic diversity. There are some other clades that have relatively few species and comparatively very little phenotypic diversity. So with these ideas in mind, one kind of following question is, are rates of trait evolution and speciation linked? And this question has kind of deep roots in evolutionary biology, with different paradigms with respect to punctuated equilibrium. Ideas of George Gaylord Simpson and Stephen Jay Gould suggest that phenotypic evolution is episodical and perhaps related to bursts in speciation and diversification rather than gradual. If we think on a more recent evolutionary timescale and consider adaptive radiations, these are lineages in which we've had lots of recent phenotypic evolution and speciation. So you can take any of your favorite adaptive radiations such as Hawaiian silver swords or Hawaiian honeycreepers, and these suggest that perhaps phenotypic evolution and speciation are linked in these particular cases. And so if we consider these ideas, oftentimes they're considered in the context of morphological evolution, and I think a lot of that is because these ideas have roots in paleontology, where morphology is king. But really, we can also think about this in the context of behavioral evolution. And so sticking with the idea of birds, if we think about bird song, it's really prominent behavior of many birds. We know that bird song is important in mate choice and species recognition, and oftentimes differences in song could present a pre-mating barrier to gene flow. And if we think about it in a population context, as in the Tibetan Plateau and the Phylloscopus warblers in this ring species system, we can have differences among populations that accumulate over time, and then when they come into secondary contact, it may be that species do not recognize each other as potential mates, and reproductive isolation has been achieved. Another interesting aspect of song is a behavior that may have some kind of macroevolutionary consequences, is that it varies in terms of whether or not it is learned or innate among different lineages of birds. And so there are some lineages of birds, such as Aucenes, hummingbirds, and parrots, that learn their songs. And essentially, this is just a form of cognitive phenotypic plasticity. And that's to say that depending on your environmental environment in which these phenotypes develop, you can have different outcomes. So one really nice example in terms of how learning and how this varies can impact trait evolution and speciation dynamics in birds is offered by the indigo birds in Africa. These are brood parasitoids that lay their eggs in the nests of other species. When the young hatch, they imprint on the song of their host, and so the females are also attracted to the song of that host. Occasionally, the indigo birds make a mistake and lay the egg in the species of, lay an egg in the nest of another species, and then those young will imprint on a different species song, and then basically reproductive isolation is achieved in that way. So that's kind of an extreme example, but if you think about it in a similar context, it may be that learning as a form of phenotypic plasticity could accelerate evolution. This is often considered something as a Baldwin effect, the idea that evolvability or phenotypic plasticity can have fairly important consequences for macroevolutionary dynamics in terms of speciation and trait evolution. And so with these ideas in mind, the two research questions that I'll present today is there a correlation between rates of song evolution and speciation rates? This will be across two groups of birds that I'll present in a second. And then the second idea is, does learning promote faster rates of vocal evolution as a form of phenotypic plasticity? And this will be a comparison between two groups of songs for which there have been kind of extensive comparative analyses of vocalizations done so far. So the two groups of birds in this study are these. The first one are the tanagers in the family Thraupidae. This is a group of about 371 species that are neotropical Aucenes, meaning that they have learned songs. They're extremely diverse in terms of ecology, morphology, basically any phenotype you can imagine. Similarly, very diverse, but also quite different in many other ways, are the ovenbirds and the woodcreepers, which are also neotropical. They have about 293 species, and they're what are called tracheophone sub-Aucenes, which means that they have largely innate song that is genetically controlled rather than developed through cultural evolution or learned through tutors. And so here are some examples of vocal diversity in the fern rise. These are wood creepers, and here's some examples of fairly typical songs from Liz Derryberry and her colleagues' publication Evolution. Many of them are trills, but they vary substantially in terms of temporal and frequency-related components among species. Historically, tanagers were considered to have fairly simple vocalizations, but there's been a lot of taxonomic revisions of what is a tanager lately, and this has led to a very different perspective in terms of vocal diversity in the tanagers. So as they're now considered, tanagers are quite diverse in song with respect to complexity among groups and the types of songs that are produced. And so since there have been some nice comparative studies done in these two groups, we have this potential to do some interesting comparisons between these distantly related groups of birds that differ with respect to how songs develop, as well as considering speciation dynamics in these lineages. So there are well-developed phylogenies for these groups. For the tanagers, we have a phylogeny of 353 species based on 6 markers that was developed as a maximum clade credibility tree. For the Fernaries, we have a tree of 285 species, also based on 6 markers. And so we combine these trees using a super tree approach with the Yates tree as a backbone. And I'll show a lot of these figures in which on the top here, these are the Fernaries. And so this part of the radial phylogeny are the wood creepers. And this down here are the throutbids or the tanagers. And so once we have these phylogenies in the super tree, we combine them with our trait data. So by combining the comparative analyses of these two kind of different groups of birds, we have 8 vocal characters that are shared between throutbids and Fernaries. Many of them are shown here, and they're basically frequency and temporal aspects of their vocalizations. Things like minimum frequency, peak frequency, song length, and things of that nature. Combined data set totals close to 600 species, and then we perform a phylogenetic principal component analysis in an attempt to reduce the dimensionality. And we also considered body mass, since it's known to be an important covariate of many frequency and also temporal related aspects of songs in these birds. We used Bayesian analyses of mixed models, which has been presented here and in other sessions already in this talk, or sorry, in this conference. So this is a reverse jump MCMC method that is used to detect evolutionary rate shifts within a Bayesian framework. And so we used BAM to acquire tip-specific and clade-specific rates of speciation and trait evolution. So we did this for speciation rate, vocal PC1 and PC2, 8 vocal characters, and body mass. So this is an example from the BAM documentation at the bottom here for the cetaceans. And really with BAM, you don't get a single answer. Rather, you get a posterior distribution of model possibilities that are sampled at different posterior probabilities. And so in this case, there's strong support for rate shift and speciation at the most recent common ancestor of the dolphins. But there are also these other models that are sampled at different posterior probabilities, just to give you a sense of how BAM works. So once we have these tip-specific rates of speciation and trait evolution, we look for a correlation between them using PGLS, using Pagel's lambda to account for phylogenetic signal and transform the variance covariance matrix, and then looked at the residuals for normality and remove any outliers as needed. And so I'm going to orient you to these figures that I'll be showing. This is the output of BAM. These are instantaneous estimations of diversification. And so, again, here we have the Fernaries and the tanagers down here. And so there are 5 rate shifts with these two groups combined. One near the root of the Fernaries and another closer to the tips. This is genus Cranioluca. Down here, within the tanagers, we have tanagers operating under a completely different evolutionary regime than the Fernaries. But within the tanagers, there are 2 rate shifts, one corresponding to a subfamily Spirophilidae, and another corresponding to Darwin's finches. And these groups will come up again later, so I just wanted to orient you to them now. But in these figures, which I'll be showing for other ones, the colors correspond to the rates themselves. And so warmer colors indicate higher rates of either speciation or trait evolution. And so there's a lot going on here, but I'm going to break it down for you. Basically, we did this for speciation rate at the top, as well as our 2 principal component axes, 8 vocal characters, and then body mass at the bottom right. And what you'll notice is there's a lot of variation among characters with respect to the degree of evolutionary rate heterogeneity that is present. So there are some characters where there is absolutely no heterogeneity. They seem to be evolving with a single evolutionary model. There are no rate shifts at all. There are others where there are a small number of rate shifts, perhaps 1 to 3. Some where there is a moderate degree of evolutionary rate heterogeneity, perhaps 5 to 10 rate shifts. And some where there is an exorbitant amount of rate heterogeneity with over 20 rate shifts included. And so once we have these kind of estimations of speciation rate as well as trait evolution, we wanted to see if there is a correlation between lineage diversification rate or speciation and these various traits. So that's what I've shown here in these scatter plots. And keep in mind that the symbols and the colors correspond to the different evolutionary regimes of the speciation rate, diversification rate. So for example, the blue circles are the background for tanagers, blue circles with yellow fill are Spirophyllinae, and etc. And so what these figures show is that we do see some evidence of correlations between rates of vocal evolution and rates of speciation. Specifically with respect to PC1, there's a positive, albeit weak correlation, a much stronger correlation between PC2 and speciation rate, vocal performance as well, and then interestingly also with body mass. Strangely, we see an inverse correlation between song length and speciation rate, but overall I think what these patterns suggest is a link between rates of trait evolution with respect to vocalizations and speciation across two very distantly related groups of birds that differ with respect to how their songs develop. And so this is kind of adding to a growing conversation about how trait evolution and speciation may be linked. There are a number of excellent studies that already exist and that are coming out, and many that are probably here going to be shown at this conference or already shown, demonstrating that rates of morphological evolution correspond to a species richness across different groups. This study suggests that behavioral evolution and macroevolutionary patterns of behavior may also contribute to macroevolutionary patterns among different groups of organisms. Okay, so now to shift gears and talk a little bit about song learning, whether or not this promotes faster rates of vocal trait evolution between these two groups. So what we did here is with BAM you can also achieve basically a clade-specific rate, and so we basically computed a clade-specific rate for each of our traits and then compared them between the Furnariids and the tanagers. And so those are shown here as well as the 95% confidence intervals in parentheses. Anytime that those 95% confidence intervals don't overlap, I've highlighted them in bold. And if they follow the prediction that basically the throughout-biz which have learned cultural evolution and development of their songs have faster rates of trait evolution compared to the sub-Aussians, those have a green check. If it's the opposite, then they have an X there. And so what you see is we have some, albeit kind of weak evidence that there is faster trait evolution in this group that has learned songs as opposed to genetically controlled songs, although it's not completely overwhelmingly convincing. And I think that there are probably many other aspects of song that are eight vocal characters did not encapsulate, things like the evolution of different notes and syllables that are kind of hard to characterize, and singing behavior such as when a bird sings, how often they sing, and other things of that nature that may also be evolving more quickly in Aussians and with learned song as opposed to Furnariids with innate song. And so just to conclude with this question, is there a correlation between rates of song evolution and speciation rates? First of all, we found substantial variation in the degree of heterogeneity among characters, such as some had no heterogeneity, others had lots and lots of heterogeneity. And we do find fairly substantial evidence that rates of song evolution are generally positively correlated with speciation rates across throat-bids and Furnariids, which are distantly related, suggesting that this may be a fairly common pattern in different groups of birds. With respect to our second question of whether song learning promotes faster rates of evolution, we have fairly limited evidence that vocal learning does promote faster rates of vocal evolution at deep evolutionary scales, but I believe that there are probably a host of other traits that may be also involved here that we can consider in the future. And so with that, I would just like to thank the many reporters that contributed to this project, and I would thank you for listening. Thank you. Sure.
I'm wondering, so with BAM, the way they, the package reconstruct continuous traits is based on Brownian, right?
Based on what, sir?
On Brownian process, Brownian motion.
Like the instantaneous rates for the tips? I'm not actually entirely sure on that. There's a lot of advanced ability there.
Right, but this, so basically did you test for example, OU versus Brownian? Because I suppose that for some of the traits, they may not evolve in the same manner.
Sure, sure. I mean, so like behavioral traits in particular often don't follow Brownian motion. And yeah, if you just compare models of character evolution, Brownian motion, or a model like OU or a Pig's lambda model with a fairly low value of lambda is often favored over Brownian motion. But to be honest, I'm not entirely sure how Brownian motion fits into how they abstract this as a type of trait.
Thank you.
Thank you.